Alright, hello everyone. Welcome to week 3. Today we're going to be covering chapter number 4. This week we are supposed to finish chapter number 4 and chapter number 5. But this first video is going to be on chapter 4. I'm going to be starting with the second half of this chapter first. So I'm going to first talk about prokaryote, eukaryotes, and then we will talk about cell itself. And then we'll talk about the cell organelles. And after that, we will talk about the plasma membrane. And then we'll talk about the transport system. Okay. After this, you will see a second video, which will cover chapter number 5, as I said before. Another reminder that I wanted to give you guys is that, so week number 4, so next week after this, you will have your first exam. And it's going to cover all the material that has been covered in week 1, week 2, and week 3. And of course, in week 3, which is this week, you have 2 inquisitive quizzes, one for chapter 4 and one for chapter 5. And for the actual study quiz, worth 15 points, you will see 1 combined quiz for both chapter 4 and chapter 5. Okay. All right. So now let's get started with our content. So guys, the first thing that we're going to talk about today is how do we compare prokaryotes to eukaryotes? How are they similar and how are they different? Okay, so both of these are cells, of course. They're just 2 types of cells. We are eukaryotes, which means the cells that our body has are eukaryotes. And then the prokaryote, the example for prokaryote is bacteria or archaebacteria. Okay, bacteria have cells which we in general are called prokaryotes. Now let's first see what do these 2 words actually literally mean. Okay, so let's see. There's a prefix pro, and then there's a prefix u, and then there's karyo. What does that mean? Karyo means nucleus, okay, or it means a central structure, so which we ourselves have what we call nucleus in the middle of a cell, and that's why our cells are eukaryotes. So prokaryotes, as the name suggests, they are, pro means before, so they are before the cells started having nucleuses. That means prokaryotes do not have nucleus. That means bacteria does not have nucleus. Okay. Now that difference is not just limited to the nucleus, but also eukaryotes have fancy structures called organelles. So they are enclosed compartments. Okay, inside the cell. So you have your big cell, and inside the cell you have tiny more compartments which do separate things. Okay, so they are called organelles. So eukaryotes, our cells, have organelles, whereas prokaryotes, which are bacterial cells, they do not have organelles. They do not have these internal complex compartments, and they also do not have nucleus, of course, because nucleus is one of the organelle. So prokaryotes miss on all these organelles. Okay, now talking about the actual diagram of the cells on the top, guys, you can see this is what your prokaryote cell looks like. So this is a bacteria. Now this bacteria, this is the actual capsule-like structure. So this is a cell, and inside the cell you can see there are different stuff in there. You can see that there's a ribosome, there is DNA. So this thread-like structure is called DNA, and then these tiny circular spherical molecules are ribosomes. Now all of this is sitting in a cytoplasm. Okay, cytoplasm is the jelly-like water-based structure in which all these different molecules are sitting. Okay, and then outside you can see this cell has this compartment. This cell is acting as one compartment, and that is given structure by this cell wall. Okay, so prokaryote, you can see they have a cell wall outside, and then inside they have a plasma membrane. You can see that there are 2 layers, right? One is the inside plasma membrane, which is also called cell membrane, and then you have the outside wall. It's a thick wall that prokaryotes have. Okay, now coming to the eukaryote cells, in the bottom you can see these 2 cells. Why do we have these 2 different types of cells? One of the cells you can see is a plant cell, and this one on the right hand side is the animal cell. So this is animal cell, this is plant cell. You can see plant and animal cell, they are much more complex than the bacterial cell. As we earlier saw, that prokaryotes do not have organelles, whereas all these structures that you see here, this one here, this one, all of these, this is nucleus, all of this, these are called organelles. Okay, these are the compartments inside the cell. So you can actually see nucleus. You can see we both have nucleus. Animal and plant cells, they have nucleus. They have most of the organelles that you're going to see are common in plant cell and animal cell. Although you can see that there are some big differences between plant and animal cells. So let's point out what differences do you see in plant cell versus animal cell. Okay, so you can see that plant cell, the biggest structure difference that we can see is they have cell wall, just like bacteria or prokaryotes, they had cell wall. I mean, of course, the composition is different on how those cell walls are made, but plant cell and bacterial cell, they have cell walls, whereas animal cells or our cells do not have any cell wall. Now, what other differences do you see? You guys know that plant cells, I mean, the plants look green in color, the leaves look green. Why do they look green? Because they have this thing called chloroplast, and inside the chloroplast is the organelle in which you have chlorophyll pigment sitting, right? So do our cells have chloroplast and chlorophyll? Of course not. Our cells do not have any chloroplast, whereas plant cells do have chloroplast. Now, when it comes to other stuff, you can see this large vacuole-like structure, this big sac. This should contain water and many other stuff. We'll talk more about that later. But then plant cells have this big, large vacuole, whereas animal cells do not have any vacuole. Okay, they have these tiny structures called lysosomes instead. Okay, now, so when you see questions like these, guys, read these questions, try to answer them, and you can also see if you cannot find their answer. Their answers are actually in their notes. So if you end the slides, if I end the slide, you can see in the very bottom you can find their answers as well. So right here, okay, you can do this to all the slides that you're looking at. Okay, all right. So what structures do prokaryote have in common? We talked about that. You see nucleus. They have, both of them have nucleus. Both of them have RER, rough endoplasmic reticulum. Both of them have ribosomes. Both of them have smooth endoplasmic reticulum. Both have Golgi apparatus. Both have mitochondria. Both have cytoskeleton. Both have plasma membrane. So there's a lot of stuff which is common. Okay, so let's see more questions. It says both plant and animals are eukaryotes, but there are differences in their cellular structure. What are those differences? And we earlier talked about those differences, like chloroplast, cell wall, large vacuole that plants have, whereas animal cells do not. Okay, so pay special attention to these type of differences and common things, because those are things that are going to be tested on your quiz as well as on your exam. Okay, all right. So now, guys, we're going to get into more detail. We're going to look into each organelles in a little more detail. As you can see here, this is your DNA. So this thread-like structure is what the DNA is. DNA is the genetic material which possesses all the information that is required for the cell to function. So you guys will see in later chapters, we're going to talk a lot more about DNA and protein. So right now, just know that there's DNA, a thread-like structure, which is situated right in the nucleus. So guys, this spherical structure is the nucleus, and inside the nucleus you have DNA stored, and you can see in the very middle it's a very dense structure called nucleolus, and nucleolus is where let me spell it out. So right here in the middle, this is where we call a structure called nucleolus. Okay, so nucleolus is where the tiny ribosomes are made. Okay, so there are small structures called ribosomes, which are very important. We'll talk about them later. They are actually made inside this, and then they are sent out into the cell for the function that they're going to do. Okay, so you have all this DNA structure, and in addition you have this nucleolus in the middle, and then you can see our nucleus is covered by an envelope. Okay, and this envelope is called nuclear envelope, and you can see in that nuclear envelope there are these pore-like structures. So you can see this structure, see this structure, this structure. So this is the spot, so you can see these ones. So this is the gateway from where the ribosomes or other molecules can pass through. Okay. All right. So again, this is your nucleus. So you can see here, it's shown here. You got these pores, and then this is nucleolus, and you can see that. Okay, and then you can also read some more detail about them here. Okay. Now, coming to a very interesting structure called endoplasmic reticulum. Now, what is this about? So you guys can see that this is your nucleus, and right outside the nucleus, it actually continues right from the nucleus. This structure extends called endoplasmic reticulum. Okay. Now, endoplasmic reticulum, all of this structure is endoplasmic reticulum, as you can see here in more detail. Now you can see that there are two forms being shown to you, one blue in color, one green in color. Now there's no color difference, of course, but the difference that exists is in their function and in how they look. Okay, so you can see that these structures are called smooth ER, smooth endoplasmic reticulum, because they do not have these structures that you can see on rough ER. So what are these tiny things? These tiny things are ribosomes. Where was ribosome made, guys? Ribosome was made inside this nucleolus. So nucleolus sent out these ribosomes. What do ribosomes do? Guys, ribosomes are very important molecules. They're responsible for making proteins. Okay, so they are the machinery that actually produce proteins. Okay, and proteins are produced by the ribosome in the endoplasmic reticulum. Okay, so if ribosome goes and sits on the endoplasmic reticulum, that endoplasmic reticulum is called rough ER. So rough endoplasmic reticulum, whereas the other endoplasmic reticulum where you do not have those ribosomes sitting, that's called smooth ER. Okay, so what are the functions? Smooth ER, as you can see here, it manufactures lipids and hormones, and it also breaks down toxic compounds. When it comes to rough ER, they're covered with ribosomes, as we talked about earlier, and then they make proteins. Okay, all right. Then coming to the Golgi apparatus, guys. If we go back to our main structure that we're looking at, see, this was again nucleus. This was ER, endoplasmic reticulum, and then this structure right here is called Golgi apparatus. It's also called Golgi bodies. Okay. So again, this is again covered with a membrane, and this actually works as a post office. Okay. Now what happens here? So what it does is, what do you do in a post office, guys? If you want to send something, send a package, you give the package to the post office, and then they're going to put a label on that, and they're going to ship it to the right place, right? That's exactly what Golgi bodies or Golgi apparatus do. Endoplasmic reticulum sends the protein. You guys remember ribosome was the one on the endoplasmic reticulum that was making the protein. So your protein is synthesized in endoplasmic reticulum, and then it's sent to Golgi bodies. Golgi bodies then put a label on them. What are those labels? Like a protein will be modified, okay? A protein will be modified where a carbohydrate will be attached, or a phosphate group will be attached, or a lipid molecule will be attached. All this happens in the Golgi body, okay? Where the protein is modified, and then it's sent to the right place, to the right destination, okay? That could be inside the cell or outside the cell. Now, how does Golgi body actually send those protein molecules? Again, it makes packages, right? So small packages. What are those small packages called? They're called vesicles. So guys, again, these are also a lipid bilayer. Guys, you can see this word coming up again and again, lipid bilayer, lipid bilayer. When we talk about the plasma membrane, we'll cover this in more detail, okay? Which is after the end, after the first half of this video. So again, these are membrane enclosed sacs, and they come from the endoplasmic reticulum and the Golgi bodies. You can see these tiny structures that are coming out. These are the vesicles. Okay, they are the actual transport. So imagine these as the UPS or USPS truck that you guys see. They actually deliver your package to your door. They're vesicles. Okay, now you can also see, guys, that there is a lysosome structure that you can see in the animal cell. Now you can see a lysosome right here. That's a is it a lysosome or a vesicle? I'm not sure. So you can see in your previous images, they're pointing this as a lysosome. So lysosome are again small sac-like structures. What are they responsible for? So they're like a trash bin, okay? They're the one that holds all the garbage inside the cell. So let's say you have a protein molecule that you do not want anymore. Your protein molecule will be sent to lysosome, and inside the lysosome, that molecule will be broken down. Now, why do you want to break it down? So just so you can recycle it or reuse it, okay? So if you don't need a protein right now, you don't just throw it away. You send it to lysosome. Inside the lysosome, you have enzymes, and those enzymes are going to break down your protein into amino acids, and those amino acids can then be recycled to make more proteins. Okay, so nothing goes into waste. Okay, so after that, guys, we'll talk about mitochondria. So mitochondria is a very, very important organelle inside our cells. Now, they exist in both plant cell and animal cells. They are called the powerhouse of the cell because they're responsible for making the ATP. Now, you're going to be hearing ATP tons of time in your next chapter 5. So ATP is adenosine triphosphate. We'll talk more about that in our next chapter. But this is the actual energy currency. Like, we spend dollars to buy stuff. If you want to go to grocery store and you want to buy something, you give the cashier $1 and then they give you something in exchange. If you want to do the same in the cell, if you want to, get something done from the molecules in the cell, you have to spend ATP, okay? And that ATP is produced in the mitochondria, okay? So again, it is made up of 2 lipid layers, so not just 1, but 2. So let me see if there's a bigger structure now. So you can see this mitochondria structure right here. So this mitochondria structure right here, you can see that there's this outer membrane, the orange color membrane that you can see, that's the outer membrane. And then there's this inside membrane, but it actually is modified. You can see it's folded into a maze-like structure. Okay, so you have 2 layers. One is the outside layer and one is the inner layer. The outer layer just stays in this shape, but the inside membrane actually folds into this structure. Okay, now this gives more space or more surface area for ATP to be produced. Okay, and we're going to see more of that in our next chapter. Okay. Right, now let's come to cytoskeleton. Cyto means cell. Skeleton is what gives your cell a structure. So these are the protein structures, like protein molecules, like actin, and they provide structure to the cell. They make the network and they form the framework of the cell. So you can see, like here, you can see this example here for a plant cell. These molecules here, filaments shown to you here, they're responsible for holding the cell together. So they give the cell the shape. Okay, they give the cell its structural support. Okay, it also actually, in some cases, like you have seen flagellas in bacterium, or in case of humans, we have sperm, which has a flagellum in the back, which enables the cell to move. All those cell movements are actually caused because of these cytoskeleton proteins. Okay, all right. We talked about vacuoles earlier, but again, we're going to talk about that again. So animal cells, again, do not have vacuoles, most of them, whereas plant cells have huge vacuoles, and those vacuoles have similar function to the lysosome. Like lysosomes were responsible for storage. Vacuoles in plant cells are also for storage of water and other stuff, okay, nutrients as well. But at the same time, they're also can contain the toxic compounds and then get rid of them by breaking them down or by throwing them outside the cell. Okay, all right, now let's see our next organelle, which is chloroplast. Again, this is a structure which is not present in the animal cell. It's only present in the plant cell. Chloroplast, again, as you can see, this structure right here is chloroplast. And inside the chloroplast, you can see there are more tiny structures inside, and those structures are the ones who are actually responsible for absorbing the energy from the sun. Okay, so they absorb sunlight, and then photosynthesis happens in the chloroplast, and that is responsible for making all the ATP and other energy carrier molecules in the plant cell. Okay, right. Now, we'll come back to these clicker questions later, but let's now go to our first half of this chapter. So if we just go to the first slide. Okay. So when it comes to cell, guys, it is the smallest and most basic unit of life. Why do we call it a unit of life? Because a cell can sustain itself. Okay, a cell, it's very small in size. Our human body is composed of approximately 100 trillion cells. That's lots of cells. That's what makes one human body. And then there can be organisms that are actually just unicellular, like yeast or bacterium. Only one cell is the organism. Or you have multicellular organisms like us, where we have more than one cell that makes the organism. Okay. All right. So that's what you can see here as well. They're showing you a bacteria, Salmonella, a single cell organism. Then you have Arabidopsis. This is a multicellular plant cell, and this is actually used a lot in genetic studies. So if you ever go into a research university and you go into a lab, there are a lot of labs which actually use Arabidopsis as their cell to study. Okay. Right, and then you can see here yeast. Yeast is again a unicellular organism, but this is a eukaryote. And then this one here, this is bacteria, so this is a prokaryote. Okay. Now, when it comes to cell, guys, cells do not always have to be like one particular shape. They can be different shapes and can have different functions. Like inside our body, we have different types of cells, right? We don't have just one type of cell. It's not just you take one cell. I mean, you take tons of same cell and you make the organism. No, you can actually, in multicellular organisms, different cells can take up different structure and different function. Like we have muscle cells, we have different cells in our heart, we have different cells in our legs, we have different cells in our brain. So this cell that you're seeing here is a neuron. So this is a cell which has been differentiated, and it has a special function, and they work as neurons in your central nervous system. Okay, right. Now, talking about the cell theory, guys, there were different theories that came up that tried to explain what we should call a cell and what we should not call a cell. So the most important property for a cell is that a cell comes from another cell. Okay, a cell should be able to reproduce. One cell will produce another cell, and that will produce more cells, and so on. The other important thing for a cell is to be self-sustaining, and it should be able to maintain a homeostasis. So homeostasis means that inside the cell, it's going to maintain a proper concentration of, let's say, sodium chloride or any other salt, and it's going to maintain that. It's going to maintain, let's say, it's going to maintain glucose. Okay, so inside our cell, there's supposed to be a fixed concentration of glucose, and it's maintained at that concentration. Okay, so they should be able to maintain that homeostasis. They should be able to divide and stuff like that. Okay, now then came these viruses, which can be confusing. Should we call them living or should we not call them living? Technically, they're not living because by themselves they cannot do anything, but they need a host. So when a virus infects the host, it can go into the host cell and use the host cell machinery to produce the molecule for its own benefit. Okay, so viruses can replicate, but inside another cell. They cannot replicate by themselves. Okay, and they don't do anything. So if they're outside the host cell, they're basically dead, not doing anything. Okay, so they're technically not a living thing. But that again always is debatable, okay, because there are different definitions for life. Right, now when it came to cell, guys, we have already seen the actual cell, a eukaryotic cell or prokaryotic cell, And if you remember, there was that cell membrane or plasma membrane that was covering the cell. And in the case of eukaryotes, you probably remember that each organelle actually had their own membrane. So what is that membrane? Okay, what is that made up of? So what is plasma membrane? It's the barrier that separates one cell from its surrounding. Okay, so imagine you have a house. You're supposed to have walls in your house, right? So if there's no wall, there's no point of having a house, right? So there has to be a barrier. So that barrier, in the case of cell, is plasma membrane. So what is plasma membrane made up of? So a phospholipid is a lipid biomolecule, and it has a hydrophilic head and a hydrophobic tail. Now, what is that supposed to mean? Okay, let's see some more figures here. So guys, this right here is one phospholipid. Okay, so in the phospholipid, you guys can see that there's this cap, there's this head on top. Let me activate the pen. So you got this pen, which is a phosphate. Okay, and this phosphate is hydrophilic. So hydrophilic means what? Hydrophilic means that it loves water. Okay, what is phosphate supposed to look like? Now, you're not responsible for the structure, but it's good to have an idea of what it is. Okay, so phosphate is P stands for phosphorus, O for oxygen, 4 oxygens with a total -3 charge on them. So phosphate is a structure that is sitting on top of this long carbon chain. These are called fatty acids. Okay, these long chain. This is a fatty acid. So 2 fatty acids. This is composed of tail, and this on top is the head, which is a phosphate group. Now this part, as you said, is hydrophilic. This part, on the other hand, is hydrophobic. Okay, now hydrophobic, phobia means they're scared of water. So this guy does not love water. It's scared of water, so it never wants to stay next to the water. Okay, now this phospholipid, since this part is water loving and this part is oil loving or lipid loving or non-polar loving, what happens is it makes a structure like this, where all the phosphate heads are going to touch each other, and all these tails are going to align in such a way that they are right next to each other. Okay, and then the same thing happens on the other side. The other side, you have all these phosphate groups, and again you have these tails. So tails are oriented towards other tails. Why? Because you see, this is hydrophobic, this is hydrophobic. They don't want any water to be in here because they don't like water. So this is how they orient themselves, to have non-polar or hydrophobic. That's the same thing. Okay guys, hydrophobic means they're non-polar. Non-polar means no polarity. And remember from our previous chapter that water is a polar molecule. So phosphate molecules, since they're hydrophilic, they love water. And imagine there is water sitting outside. Okay, so they're very happy with each other. On the other hand, this is non-polar, this fatty acid. So that's why they hate water and they do not want to orient themselves towards the water. And that's why the tail is pointed towards inside and the head is towards the outside. And then same is with this molecule. So there's water here as well. So imagine this whole thing is surrounded by water, and that's why all the phosphates are outside and the tails are inside. Okay, and it makes a structure like this. Now if you see this in 3D, this is what it's going to look like. Inside there's water, outside there's water, and then this is the phosphate head, and this right here is the lipid membrane. Okay, so I mean, this is the head of the membrane and this is the tail. So this, if you look at the phospholipid, this is the phosphate group, this is the lipid, this is the fatty acid group. So there's this one on the top, so this top one, and then the bottom one is right here. So you can see how it makes a bilayer of a phospholipid. So phospholipid is this thing, and then there are 2 of them, so 2 layers, so bilayer phospholipid. Okay, when a phospholipid bilayer comes together. So see, if you take phospholipid bilayer and you put it into water and you shake that, it's going to turn into this structure called liposome. Okay, so the some means body and this is lipid body. So what happens is you put these individual molecules, tons of them, and just put them into water, shake them, it automatically gives you this shape. Okay, that's amazing. And this gives you the idea of where the cell probably came from. Okay, imagine there was DNA, there was protein, and all these molecules, and then phospholipid came in somehow, and then they came together. So all the phospholipid made this wall-like structure, and it gives you like that compartmentalization. So and then DNA and your protein, everything is sitting inside. So you're basically, this is what a cell would look like without any of its organelles. So without any stuff inside, this is just showing you the outside envelope. Okay, so that's what liposome is. Liposome is everything that the cell is minus everything inside the cell. Okay, so only the envelope of the cell. Okay, so yeah, I think we covered whatever these slides are saying. Okay, now there are some questions that you should be looking at. Why is it important that phosphate head of a phospholipid is hydrophilic? So it's asking you basically, why is it important that the phosphate group, so this group right here, which is the head of phospholipid, is hydrophilic? Why is it important for it to be hydrophilic? You see, one or the other part of the membrane has to face water. So if this will be polar, that's the only way this can face water. Okay, that's why it's important to have one side which is hydrophilic, the other side which is hydrophobic. That's what is going to give you a wall-like structure, a membrane. Okay, all right. Again, now this is asking you, what does a liposome lack compared to a cell? Of course, does not have DNA, does not have nucleus, does not have ribosomes, it does not have anything that a cell inside has. Okay, all right. So now again, we talked about this already. It's saying, do you think phospholipid bilayers would have played a role in origin of life? Yes, maybe this is how the cells originally came into existence, right? That this was this compartment-like structure that was made, and inside that all these different biomolecules entered. Okay, or there was biomolecules that were engulfed by the liposome, and that's how the first cell was made. Okay, maybe. Okay, now when it comes to plasma membrane, we gotta talk about its permeability, its selective permeability, and the transport. Okay, so what you can see is if you have a cell, that cell, the stuff has to move inside the cell and outside the cell. So we have different molecules. Let's say, for example, glucose. When you eat food, when it's digested in your stomach, when it goes through your intestine, the cells have to absorb that glucose and bring it into the cells, right? That's how a glucose molecule is going to go from your food into the cell. So if a cell does not have any way for that glucose to come in, glucose will never come in. So what do we need? We need gate-like structures. Okay, so first of all, what does selective permeability means? Permeability, of course, means that it's going to allow some of the stuff to come in. Selective means that it only allows some of the molecules to come in. So it's not like an open door that everyone can come in. Membranes, and they are selectively permeable. They will let some things pass through, the other things won't. Okay, now when the molecules want to move through that bilayer, phospholipid bilayer, we need proteins that need to be situated in those membranes, so they're embedded into the membrane so that things can pass through. Okay, so they act like tunnels from where you can pass the things, okay, pass the molecules. Now, there's this two important categories of active transfer and passive transfer. So let's quickly talk about that, okay? Let's talk about now actually I wanted to show you guys the actual phospholipid, how would it actually look like. So if you guys remember that phospholipid structure that we were looking at, there was that cap on the top. So there was this, this is that head, okay? And this is that long fatty acid chain that we're talking about. So basically, what is this? These are all carbons. Okay, so the long carbon chain. This is the part that hates water, hydrophobic, and this is the part that loves water. You see, it has charge on it, and water loves charged molecules. Okay, so this is your 1-phospholipid. Okay, all right. Now let's talk about the transport. Okay, now your transport is basically, let me select the pen. Okay, so your okay, what's wrong? Give me a second, guys. Okay, so the transport is basically 2 types. One is the active type, and the other one is the passive type. Now, as the word suggests, guys, active means it's gonna use energy. Okay, it uses energy. Passive, on the other hand, means it's gonna work without the use of energy. Okay, so no energy required. Okay, now in passive transport, what happens is, let's say glucose has to move from its high concentration to a lower concentration. So it has to move from one place where there's a lot of glucose and wants to move to the place where there's less glucose. That's going to be a passive transport. Okay, it moves with the concentration gradient. So with concentration gradient. So now you can tell that the active transport, on the other hand, is going to be opposite. It actually goes against the concentration gradient. Okay, against the concentration gradient. So let's quickly draw a diagram and see what that should look like. So guys, imagine that you have this container which is divided by this membrane, and then we're considering that this membrane can let things pass through. Okay, so imagine that there are glucose molecules here, and there are lots of glucose molecules on the left hand side, and on the right hand side there are very few glucose molecules. Okay, so we'll say this is the low concentration site, this is the high concentration site. So if you want to move stuff from the left hand side to the right hand side, that's going to be easy, right? See, there's high concentration, it very easily moves to the right hand side. That's what passive transfer is going to be. Okay, but when it wants to move from against the concentration gradient, so when it want to go from low concentration of glucose to the high concentration of glucose, that's hard because you're going against the concentration gradient. This is like swimming upstream. Okay, against the stream. This has to use energy, so ATP is used. So there will be some molecule structure sitting here that will help you transport your glucose from here to here against the concentration gradient. Okay. Now, for passive, of course, the way it's going to be is here the movement was going from right to the left. You're going from low concentration to high concentration. That was active transport. When we talk about the passive transport, again, if we see that there is a high concentration on the left hand side, and this time we're going to move from the left hand side to the right hand side, this is going to be passive transport. Okay, this is going with the concentration gradient. You're going from top of the mountain to the low of the mountain. That's going to be easy. Okay, but on the other hand, when you're going from bottom of the mountain to the top of the mountain, that takes energy. Okay, so passive transport, it just goes with the concentration gradient. Passive means it's not going to take any energy. Okay, but when it comes to passive transport, guys, there are 2 different types. Okay, when it comes to passive transport, now again, let me introduce this word to you guys. When you go from high concentration to a low concentration, you're basically diffusing your molecule from one side to the other side. So this process is called diffusion, okay? Diffusion. Now this diffusion can happen in two ways. One is simple diffusion. Okay. The other one is facilitated diffusion. Now let's see what is the difference, okay? Now as the word suggests, simple means it does not need any fancy equipment. Where there's facilitated diffusion, it's gonna need something on this membrane for things to pass through, okay? So simple diffusion, guys, is going to work for molecules which are very small. So let's say a water molecule, or CO2, carbon dioxide molecule, or oxygen molecule. These are very tiny molecules. They can just pass through this membrane without the help of any transporter or any channel or any gate, okay? On the other hand, for facilitated diffusion, even if you are going with the concentration gradient, you're going from high concentration to low concentration, it is still a passive transport, but you need some transporter, you need a gate here, okay? So stuff like Na+1 or K+1 or calcium, or other big molecules, let's say glucose, okay, or maybe another protein molecule, okay. If you want to move any bigger molecule, or you see these guys, charged molecules, they cannot just go through the membrane. They need help from somebody. That's why it's facilitated, and then they need help of a transporter, okay. So they need some kind of gate or channel to be present here, okay. Now you might have noticed here that here we actually have H2O, we have CO2, we have oxygen, which are just passing through the membrane without any problem. On the other hand, you have smaller ions. You see sodium, potassium, calcium. Sodium is smaller than CO2 or H2O or oxygen, O2, because sodium is just one atom. These are 2 atoms, 3 atoms, 3 atoms. These are actually bigger than sodium, potassium, and calcium. But why are sodium, potassium, and calcium not able to pass through the membrane? Now, the important thing here, guys, is we'll go back to our nice structure of our membrane. So this is what makes a membrane, right? This is our membrane. So what we're saying here is that the water can just pass through. So you take H2O molecule, and it's just gonna pass through this membrane without any problem. It does not need any help from a gate or a channel. On the other hand, or even for CO2 or O2, they can just pass through, right? But when it comes to stuff that has a bigger size, of course, and if it has a charge on it, it cannot just pass through the membrane. Why not? You see this membrane carefully? This was the polar side, so this guy loves water, right? But this side is non-polar. Now, non-polar means fat, right? Fat means it hates things like water. It hates things like charged particles or anything that is polar, right? So if you take Na+, now Na+ has a charge on it. So charged particles are happy with water, but they're not happy with fat. Okay, so a charged particle will not be able to pass through this membrane. Okay, that's why it needs a special structure. So let's see those structures. These are those structures, guys. You see this structure, these 3 structures. Now, what's happening here? Let's look at this figure more carefully. Okay, so now this figure is showing you a few different things. One, on the left hand side, it's showing you that there's active transport, and on this side is showing you passive transport. And as we learned, passive transport is simple diffusion or facilitated diffusion. Simple diffusion means a molecule like water or CO2 or O2, very small tiny molecules, can just pass through the membrane. Okay, now you might be thinking water is actually a polar molecule and our lipid here is non-polar, so things should actually not be able to pass through the membrane, but water is so small that it passes through. Then you will say, okay, carbon dioxide and oxygen, what about them? CO2 and O2 are non-polar molecules and they can technically pass through them because they're nonpolar. So they can just diffuse through this membrane, okay? Now, when it comes to things like ions, which are actually smaller, but you see they have a charge on them, Ca+2, K+1, or sodium +1 you see here, they all need some opening. They need a channel, they need a tunnel, they need a gate that will make them pass through them. Okay, so what else is going on in this figure? This is passive transport, guys. What does passive transport mean? Passive means no energy requirement, means it's going to go from top hill to the bottom, right? So it's going to go from high concentration to low concentration. So look at look at water here. Look at water here. You have tons of water molecules. Here you have very few. So water molecule can just pass through. It can just go from here to here very easily just by diffusion, okay? It's going to be a passive transport, does not need any energy. Now you look at potassium, lots of K+, very few K+. It goes from high concentration to the low concentration. You can see the arrows pointing inwards. So potassium, sugar, calcium, all are moving inside. They're going from their own high concentration to their own low concentration. So this is passive transport. Okay. Now, when you come to this sodium, on the other hand, you see there's tons of sodium here, and then there's very few sodium here. Now, what is different in this structure? What is this showing you is that sodium wants to actually move outside. Okay. So your sodium ion is moving from inside to the outside. So it's actually going against the concentration gradient. Sorry, guys. Here, there are lots of sodium ions. We don't need any more. But cell decided to move some sodium ions from the low concentration to the high concentration, going against the concentration gradient. This will require energy, ATP, and that's why this is active transport. Okay, again, guys, just to review this, active transport means it's going against concentration gradient. It's going to need energy. Passive transport means it's going with the concentration gradient. It's going from high concentration to the low concentration, so it does not need any energy. But there are two scenarios. One is simple diffusion, one is facilitated diffusion. Simple diffusion, you do not need any channel or tunnel. On the other hand, for facilitated diffusion, you need these channel-like structures for molecules to pass through. Okay? All right. Remember the example, guys, which ones need channels, which ones do not need channel. You will be tested on that on your exam and quiz. Okay. All right. So in what ways is plasma membrane a barrier, and in what ways is it a gatekeeper? So see, it's a barrier because it's not letting everything come in. Why is it a gatekeeper? Because it allows selective things to pass through, not everything. Okay. Now, why can't ions like Na+ cross the plasma membrane without the help of transport protein? So guys, saying that sodium ion is so small, so why is it not able to pass through? We already talked about that sodium plus has a charge on it, and these type of ions, molecules, cannot pass through without the help of transport protein, because these are charged molecule. Our membrane is non-polar. Okay, it hates stuff like these who have charge, so it won't allow them to pass through. If no energy were available to the cell, what form of transport would not be able to occur? What forms of transport could occur? So guys, as we learned, passive transport does not need energy, so that will keep working even if there was no energy. Which one will not occur? Active transport will not occur. So anything going against the concentration gradient is not going to happen if you don't have energy. Okay. This is talking about diffusion, guys. What does diffusion means in general? I already talked about this. So diffusion is just going from high concentration to low concentration, or basically in this example you see that you're adding a food color from a highly concentrated one region. It's diffusing to the rest of the place, and it finally reaches this equilibrium position where all the water is reddish color shaded, because that this food color was able to spread out evenly and make a homogeneous kind of solution. Okay. All right. Okay. Now, guys, osmosis, again, a very important concept. Osmosis, guys, has to do with water movement. Okay. How does water move from our cell membrane? Of course, we learned that it does not need any transporter, but still, it is a special diffusion. It's a simple diffusion, but for water, it's a special case called osmosis, where water moves from its own high concentration to low concentration. Okay, let's see, what do we mean by that? Okay, so guys, again, if we try to draw our container, and then I tell you that there is a semi-permeable membrane. A semi-permeable membrane means it's permeable to only few things and not all. Okay, so imagine that there is again glucose molecules sitting on this side of the membrane. So guys, semi-permeable membrane is similar to our plasma membrane. Our cell membrane is also semi-permeable. It allows water to pass through but does not allow other molecules to pass through. So there's glucose on this side, and then there's a little bit of glucose on this side as well. But then there's lots of waters. Okay, so lots of water on this side. And then if we talk about the left hand side, see, this is glucose in water, so of course there is some water. Okay, the whole point is, of course, guys, do not go by the number of water molecules that I'm showing you here, but the point is that here it's less concentrated glucose on the right hand side, and on the left hand side it's a high concentration glucose. Okay, so what you can say here is that there's high solute. Remember guys, solute and solvent. Solute is the one that you add, solvent is where you add. So here there's high solute, here on this side there is low solute. Okay, let me erase this top part. Okay, alright, so this is the low solute region, so there's less glucose here, and here there's more glucose. So we can also look at this with the perspective of water. Here, the low solute concentration means a lot of water. A high solute concentration means very little water. So where does water move? Because in semi-permeable membrane, or in the case of osmosis, who moves? Can glucose move? No. Who moves? Only water moves. Okay, where does water move from? What do you guys think? See, it's a simple diffusion, so it's supposed to go from its high concentration to low concentration. That's exactly what happens. But listen to this carefully. I'm saying it goes from water. So water goes from its own high concentration to its own low concentration. Okay, so do not get confused. Don't confuse that with low solute concentration and high solute concentration. Okay, your focus right now is on water. Water is the one who is moving. Okay, so water will move from where there's more water to where there's less water. So on the right hand side there's more water, on the left hand side there's less water. So water will move from our right hand side to the left hand side. Okay, because it's moving from high. So water moves from high water concentration to low water concentration. Or if you want to think about this the other way, what you can think of is that the water moves towards higher solute, so in this case glucose concentration. Okay, so what is basically happening is water is moving from low solute concentration to the high solute concentration. You can think of it this way, or you can think of it as water is moving from its own water concentration, high concentration, to the lower water concentration. Okay, so it just depends on what you pick, on what you're thinking about. Okay, all right. Okay, so again, this does not require any energy, and only water is the one who is moving. Okay, so here again, this is the same figure that I just drew for you. They're showing you sugar molecule and water molecule, and this is semi-permeable membrane, and water is moving across. It's moving from right hand side to the left hand side because left hand side has glucose. Right hand side either has little glucose, the structure showing you that it has no glucose at all. So your water is the one who will move from high water concentration to the low water concentration. Okay, all right. Now we got to talk about these three different cases: isotonic, hypertonic, and hypotonic. What's going to happen into each case? So if you take water and you put it into different salt concentration or different glucose concentration, different solute concentration environment, then what is going to happen to the cell? Okay, so make sure you guys listen to this carefully as well. So what's going on here? Okay, so you're basically have 3 different containers, and we're going to call the first one isotonic. Then the second one is hyper. Hyper means high. So tonic, like as in salt concentration, this is high salt concentration. And hypo means low. So hypotonic. So hypertension, like high blood pressure. Hypotonic, low blood pressure. So hypertonic means a high concentration. Hypotonic means low concentration. Isotonic means same concentration. Okay. So what we're saying here is that we have a cell in each case. We take same cell, we put them into 3 different solutions. Isotonic means, let's say we're talking about salt concentration, okay? So we're talking about NaCl concentration. So if inside salt concentration is 1% in the cell, and outside this is also 1%, we'll call that isotonic solution. Where will water move? Water is not going to move because the concentration is same outside and inside. Okay, so even if the water is moving, equal amounts of water will go outside and equal amount of water will come inside. So in net, there's no movement. Okay, all right. When it comes to hypertonic, what does hypertonic mean? Again, we're going to assume that inside the cell it's still 1%. So in all 3 cases, inside the cell it's 1% NaCl. Okay, so we're saying that inside the cell it's 1% NaCl in each case. But when it comes to the solution where we're putting these into, hypertonic, again, here, let's say it's 10%. Okay, that's a lot of salt. So from you take 1% cell, 1% salt concentration inside cell, you put it into 10% sodium chloride solution. What is going to happen to the cell? Where will water move, guys? See, this is high salt concentration. There's too much salt here. Okay, so where do you think water is going to move? Remember, water will move from its own high concentration to its own low concentration. See, outside there's too much salt, inside there's very little salt. So this water inside the cell will want to move outside. You guys see that? Why? Because see, water inside is at high concentration. Outside there's very little water, so it wants to compensate for that water and it comes outside. So what's going to happen to the cell, guys? The cell will shrink because water came out. What's going to happen in the hypertonic? So guys, in hypertonic, imagine this is 0.1%. Inside the cell, 1%. Outside in the solution, it's 0.1% NaCl. Okay, the solution is 0.1% only, so very diluted. So now what we're saying is that there's less salt and there's more water outside compared to the inside. So now what's going to happen to the water, guys? The water will go from outside to the inside. So again, with these arrows, guys, I'm showing you the water movement. Okay, we're talking about osmosis. So this is water's movement, not salt movement. Water is the one who moves, not the salt. Okay, so water is now going to move inside. Why is it going to move inside, guys? It's going from high water concentration. There's too much water here, too little water here. So water wants to go inside. Now what's going to happen, guys? The cell will either expand or is actually going to explode. So your cell will explode because of the high water amount which is going in. Okay, so of course we want our blood to maintain isotonic conditions to what it is inside the cell. We do not want our blood or other fluid concentration, salt concentration, to be either very high or very low, because that's not good for our cells. We want isotonic conditions, right? Okay, let's see. I think that was oh yeah, there's one more topic that you got to cover. What is this saying? Okay, so how can we move things across? Now, one way, of course, that we learned was we learned the active transport, passive transport. We learned the simple diffusion, active, simple and facilitated diffusion. Then we talked about our water osmosis. But what is the other way to move the thing across? What if we want to move a big, huge molecule from outside to inside or from inside to the outside? How is that going to work? So that works, guys, by exocytosis or endocytosis. And how does this work, guys? This works again as the vesicles. So vesicles, again, we have seen that before in the beginning of this chapter, that there are these plasma membrane circular structures that contains the molecule that you want to get rid of, or the ones that you want to bring in. So they're the actual transporter, right? So they're like that UPS truck, right? So exocytosis means outside the cell, and endocytosis means to bring into the cell. So exocytosis is used for exporting the cells out, exporting the things outside the cell, okay? And endocytosis for bringing the things into the cell. Now, why do we need this again, guys? As I mentioned before, if you have a huge molecule, you need stuff like exocytosis, endocytosis. If you want to move particles that cannot come through a channel or transporter, we need all these vesicles to move, okay? And then this endocytosis, guys, which is to bring in the stuff, this can be specific or non-specific. Non-specific means that that all the material can pass through. Specific endocytosis means a particular molecule which has a tag on it is the only one who will be able to come in. Okay, so that's a specific endocytosis. Now, receptor-mediated endocytosis, guys, this is that specific endocytosis. So let's say you have a cell, and inside that cell, guys, you want to bring in something. The thing that you want to bring in has to be tagged with a molecule which the receptor on the cell membrane is going to recognize, and will only let that molecule come in. Okay, now let's talk about phagocytosis. What is phagocytosis? It's also called cellular eating. This is non-specific. So is pinocytosis. That's also non-specific. Phagocytosis, guys, means it is the endocytosis of big things. Okay, let me show you a quick picture here. So, guys, this is endocytosis. What is this blue structure? This is one cell inside our body called macrophage. This is a white blood cell, guys, and this red cell right here, this is a yeast cell. So if we got a yeast infection or something, this macrophage, or one cell, is going to eat this yeast cell. Okay, so this is not going to happen by diffusion or by a transporter protein, of course, right? Because that's a huge molecule. I mean, huge substance. This needs the whole vesicle, the whole plasma membrane to open up and engulf this thing inside, okay? And that's endocytosis. And this specifically is called phagocytosis because this is for big stuff, okay, like a cell. Pinocytosis on the other side, guys, also non-specific, but it's for liquid. So this is like drinking. On the other hand, phagocytosis is like eating, okay? All right. So let's see these questions. If endocytosis itself is non-specific, how does receptor-mediated endocytosis bring on these certain molecules? Again, as I talked about this before, guys, you can read this part here. That receptor-mediated endocytosis is a selective process in which certain molecules will come in, okay? Because they have some kind of tag on them. Like, for example, our liver cells have a receptor called LDL receptor, and they only take in LDL. So LDL, you guys might have heard of LDL, low density lipoprotein. Also there's HDL, high density lipoprotein, or VLDL, very low density lipoprotein. All these, if you've got your lipid profile, or you've got your blood tested for your cholesterols or triglycerides, or LDL, that's what they look for. So these molecules, these lipoproteins, they need to be transported inside the liver. So liver cells are supposed to have a receptor for them. So a receptor is like a sensor that is on the liver cell. If it senses LDL, it's going to open up and let the LDL come in. Okay, so that's how that specific endocytosis work. Okay, so it says what sort of molecule will move by endocytosis, exocytosis? Again, the bigger size molecules, okay, which cannot go through just by diffusion. Okay, and then again here it's saying, third question says, how does fluid that enters a cell via pinocytosis differ from the fluid that enters by osmosis? Now you can see osmosis, remember, was only water movement from water's high concentration to the low concentration, whereas pinocytosis, guys, was just moving in non-specifically any solution. So if the fluid outside the cell, outside the cell membrane, has salt dissolved in it, or glucose dissolved in it, or something else, if that vesicle opens up, it's going to bring all of that solution inside, not just water. Okay. Okay. Now, guys, for this, that's the end of our chapter. But I quickly wanted to show you guys the endocytosis and exocytosis figure real quick. I saw this picture. You can see here, this is showing you exocytosis, so stuff inside the cell is being thrown outside. And this is endocytosis, cell from outside the cell, stuff from outside the cell needs to come in. So you can see this is the vesicle that has been formed. So this is your truck that you prepared. This is the UPS truck that needs to be sent out. Okay, so you can see that this is made up of lipid bilayer. And you see, since this is lipid bilayer, this is also our plasma membrane, which is also lipid bilayer. They can actually diffuse into each other. And see, when this happens, you see this vesicle, this is your same vesicle which has become part of the plasma membrane. And since it opened up, it was able to put all these packages outside. Okay, so that's exocytosis. Endocytosis, on the other hand, is just the opposite. What happens is your plasma membrane changes its shape. So it's trying to engulf all this stuff inside. So it changes its shape like this. It forms this vesicle-like structure. So first is just the bend. Things come in, and then you can see it pinches out. It pinches out and becomes this complete vesicle, and this can now just go anywhere inside the cell. Okay, so pretty much, guys, that's the end of this chapter. I'll be posting another video which will be covering chapter number 5. Okay, and guys, again, I said before, please listen to these lecture videos. This will guide you on what you need to know for your study quizzes and for your exam. I hope these will be helpful. Okay, see you guys next time.